皆さん、こんにちは。時間になりましたので、セミナーを始めさせていただきたいと思います。私は、バイオサイトジェンの日本のエージェントをやってます、角村と申します。どうぞよろしくお願いいたします。今日は、皆さんお忙しいところお集まりいただきまして、本当にありがとうございます。今日のセミナーはですね、弊社の、チーフサイエンティフィックオフィサーのベニー・ヤンの方から、最初に、抗体、100% 人の抗体を発現させるモデルを使いまして、その、抗体の、いろんな種類の説明ですとか、活用の方法ですとか、そういった抗体の方のご説明をさせていただきます。で、次に、シニアプロダクトマネージャーのチャオからですね、難しい。同じく人かマウスのモデルを用いました、BBB を、通過して、で前臨床ですとか、神経関係モデルの活用するような内容についてのお話をさせていただきます。で、申し訳ないんですけれども、弊社のメンバー、日本語喋れる者がおりませんので、セッションと質疑応答も全部英語になってしまいますんで、すいません、そこはご了承ください。で、途中で、プレゼンテーションの途中でも質問とかありましたらいつでも止めていただいてご質問、していただいて結構でございます。それからご紹介というか情報なんですけれども、弊社、今日本の皆様の、ご注文もたくさんいただいてビジネスがだんだん大きくなってきたものですから、日本で、今年にオフィスをどこか立ち上げようということで考えております。ちょっとまだ場所は確定はしてないんですけれども、ここの iPark とはちょっと契約をしまして、で、もしかしたらここの中でオフィスを作るか、またちょっと別のところになるかもしれないんですけれども、で、近々、日本語を喋れる、スタッフを、雇用してですね、常駐になるかどうかはまたあれなんですけれども、もうちょっと日本のビジネスを力入れて展開していこうというふうに考えております。ですから、これからもうちょっと皆様方にも、より良いサービスをご提供できるようになるかなというふうには思っております。あと弊社、本社とか動物の施設とか全部もちろん中国なんですけれども、最近中国に訪問するにあたってですね、ビザがいらなくなりまして、普通にチケットの手配で、中国の方にも来ていただけますので、今後もし何か機会がございましたら、ぜひ弊社の方にもお越しいただければ幸いでございます。では、早速ですけれども、セミナーを始めさせていただきます。Okay, now we can start.
Yes. Good afternoon, everybody. Welcome to this Biocytogen seminar. I'm Benny Yang. I joined Biocytogen in 2016. So I'm training in the United States, and then after I come back to the Biocytogen, I'm leading the antibody discovery part. So today, my colleagues will give you an introduction about Biocytogen's business and see how we can help your research and the drug discovery. So I think first I'll give you a little bit background about Biocytogen. Biocytogen is a company founded in 2008 in Massachusetts, United States. Today we have become a global medium-sized biotech company. We have overall over 1,000 employees in different locations. Our headquarters is in Beijing, and we are also doing a lot of research. And we also have a site in Shanghai. We also have a facility in Boston, in San Francisco, in San Diego, and in Heidelberg, Germany. So we have a lot of presence in the outside of China because a lot of our partners and our clients are in the United States and in Europe. Biocytogen is also a public company. we are traded in Hong Kong stock market. So our business can be divided to two different lines. And on the left you will see we call them biologics. RenBiologics is about how to discover the fully human antibodies using a series of novel transgenic mice. But on the other hand, we also have another business branch called biomice, and my colleagues will introduce that. So these are the humanized mouse models, over 1,000 of different models for screening and testing different antibody therapeutics in those models. And these models are welcomed by a lot of companies, and all top 10 MNCs are using biomice for their research. But then for the first part, I will introduce the RenBiologics. So Ren means human in Chinese characters. So RenBiologics is about how to discover fully human antibodies. So there's different ways to do the antibody discovery, but we mainly using the in vivo approach, which means by immunizing mice, we can generate the binders against the different targets, then using those binders for drug discovery and development. But ideally, we want to have the fully human antibody. That's why we engineered several different mice strands, and these mice will produce fully human antibody after you just giving immunization. So besides these platforms, we also did a large campaign for doing the ready available antibody binders for our clients and collaborators. This is because a lot of times if you are working with antibodies, right, you don't want to go to CL company and to ask them to do like immunization campaign, and which takes a lot of time, and also most times it's probably not successful. But what we want, as many companies and many they're working on different like modalities. They want to use the antibody as a drug or as a way to deliver the drug to the target areas. So they're not specialized in the antibody discovery, but that's why I think we can help them to get the best antibody to help them help their research. So in the last 3 years, we did a large campaign. So we choose more than 1,000 targets. And we did immunization for every individual targets, and we generate a large binder library. Currently, we have more than 1 million fully human binder for these targets. And these binders are not just, we just have a sequence. We also have all the functional data, characterization, like affinity, binding to the cell, binding based on cell-based assays, and the cross-species reactivity, like whether they can bind, not only bind to the human, but also bind to the monkey, bind to mouse, bind to rat. So these are the early stage binders. They can be further selected and developed into a real drug for human clinical trials. And internally, we are doing some like selection, but we also are doing a lot of external partnership. And we are helping along with our partners, discover bispecific antibody, ADC molecules, also using the nanobody-derived sequence to deliver the drug or making the cell-based therapies. So our business model is very flexible. So we have, since we can provide readily available sequence to our partners. So this is really welcomed by the whole community. And in the last 2 years, I think we have completed more than 200 assets-based licensing. Basically means we provide our partner with a sequence, and they can further doing the testing and selecting the PCC molecules. And we can also sometimes we also licensing our technology platform to our partner, and they can do their own propose their own targets for antibody discovery. So I will first introduce the mice, how this mouse is made. So this shows why today, like if you're working with antibody, and you know antibody in the past we think it's just a drug, but today it's not just a drug. It's also a way to deliver the drug to different and combine with different technologies to build more new modalities. So antibody can combine with small molecules, large molecules, can be combined with mRNA, combined with cell therapies. So, but overall, whatever you do, antibody is still lying in the center of this cartoon. So when we talk about antibodies, when we do the selection, so we always know we want to have a high affinity. That's the criteria for good antibody. And also, for a large molecule, you usually will have different epitopes on the surface of the target. So usually where the epitope, the antibody binds, is very critical for its function and for all the relevant successful of the target project. So we want to have easily to generate a larger pool of antibodies, which can have bind to different epitopes, means we can generate a diverse pool of library. And we want to have the fully human antibody because this will reduce the immunogenicity when you test the drugs in the human patient. And what we want here, to a little bit different from other companies, is we are providing a readily available library for this more than 1,000 targets. And these are the library bind to all the targets in different indications. So this will make the selection or the building of new drugs much easier. So I would here introduce the RenBiologics. So RenBiologics including a series of novel transgenic mice for different purposes. So on the very left one, you see a platform called RenMab. RenMab is the mouse to generate the fully human monoclonal antibody. So in this mouse, we replace the variable genes of the mouse heavy chain and light chain locus. So we replace the coding sequence with the human counterparts. So this mouse can generate the antibody response against the immunized antigen. But the sequence you get retrieved from this mouse is already fully human. So you don't need to do any more humanization work. Sometimes we want to build the bispecific antibody, and that's really challenging. And also here, so what do we think? There's a very, very powerful way to do that is building a 1+1 format bispecific antibody using the common light chain solution. So we generate a mouse which we fix the light chain sequence, and all the heavy chain, all the sequence antibody in this B cells in this mice will have different heavy chain sequence, but they share the same light chain sequence. And once we generate the antibodies, we can easily assemble the binders from two different campaigns into one molecules by co-expressing two different heavy chains for different targets, but since they share the same light chain, so we just co-express with the light chain. So when you co-transfect the two heavy chain and one light chain in the same cell, it will automatically assemble into a bispecific molecules. And we have shown this yield a very high production, and also the molecule has very good physical chemical properties. In the middle, you will see the VHH format platform. This is the RenNano. So RenNano is the mouse to generate the heavy chain only antibody or nanobody. So this is very similar to the heavy chain only antibody generated from a llama or a camel. But here, what we can do is generate the antibody from the mice, and also this sequence are fully human. You don't need to do humanization, so which is tedious, and sometimes it's still not very good for drug development. And also, if you're interested in ADC development, we also have a platform called bispecific ADC platform. So here we combine our bispecific antibody platform with our unique linker payload, and I will show you some. We have some assets at the clinical stage, which we partner with another company. Besides the conventional antibody against extracellular targets, we can also target intracellular targets like HLA peptide complex. So this can be done by two different fashions. One is we can generate the fully human T-cell receptor binders against the intracellular targets. So this is called fully human TCR mouse, RenTCR. The other mouse is called RenTCR mimic platform. So similarly to the RenTCR, but this time we can generate the HLA peptide binder using the antibody format. But sometimes if you have difficult targets like a GPCR, which is notoriously difficult to generate good binders, we have a platform called GPCR platform. So these are the way to generate a good binder because we can knock out the individual gene on the mass background. So this will ensure you get a much robust immune response, and then you have higher chance to retrieve the good sequence. So with all the three major tools, the RenMab, we can easily generate the fully human antibody if you are just doing generating monoclonal antibody. But if you are interested in bispecific antibody, we can do two different ways. We can either using the light and immunize with two different antigens, and we can assemble this sequence into one molecule using the novel interval approach. So this is one way to generate 1+1 format bispecific antibody. But the other way is using the RenNano, because the RenNano does not have the light chain. So only with heavy chain sequence, you can easily assemble them into multi-specific antibody. So our platform, all these mice are generated with the new technology. So we call it in situ replacement. So this is very different from the old method, which they tend to use in the knockout approach, knockout the endogenous mouse B-cell gene locus, then put the human human antibody gene sequence as a transgene. This can be done, but it's not stable, and also it's not. Those mice really usually you encounter like a compromised immune response. But when we do the in situ replacement, because this all the B-cell development is using the endogenous regulatory elements, so the B-cell develop much healthy, and also you will get a much better immune response, and also you get as a result you will get a much better antibody sequence from these mice. And our mice have been recognized since we launched these tools back in 2020. So these have been really also many big companies also licensing our technology for their internal research. So one additional trick for generating good antibody is using a target knockout approach. We know human and mouse usually have shared a very high homology of the genes. So if you're just using mouse immunized protein to mice, usually you will not generate a very good immune response, because if the areas you're interested in is highly conserved between mouse and human, so this will usually the immune response will not bias toward those regions. But we think we can overcome this problem by using the target knockout approach. So if you can knock out the individual gene on the RenMass background or other background, you will generate much better response. And this time, because you don't have to overcome immunotolerance, so you have the antibody library you get will be directed towards more diverse epitopes. So we took this approach to generate the antibody, and we are not we apply this not just to one single target. So we select more than 1,000 targets, and we are doing the target knockout approach first, and generate different mouse lines, and using those knockout mice for antibody discovery. So we have been doing this for several years, and this has been very successful for our antibody discovery campaigns. So currently we have many binders for different targets, and if you are interested in, you can check our website, which is shown on the bottom right. But basically, if you look at the website, you can search your target by different ways. One method is to look into the mechanism of action to see what's the biology of this target. Then you can further zoom in that category. But the other way is to look in the therapeutic areas to see whether we already have your interested targets in our database. So in the last several years, we took a lot of effort to generate a large number of antibodies. So these antibodies encompass the targets for oncology, for inflammation and autoimmune disease, for neuroscience, for metabolic disease, for cardiovascular disease, and for viral infection. Basically, we're generating the antibody for all different implications. And this overall, because some of the targets are overlapped, because like some like for the immune-related targets, this can be used for both oncology and for the autoimmune disease. But overall, when we add all the numbers together, there's about around 1,000 targets. And for these targets, we generate more than 1 million fully human binders. And we not just only have the sequence, but we also have all the initial characterization data. So we know the affinity and binding specificity and the cross-species reactivity for every single sequence we have in our hand. So this really makes the selection and building more complex drugs much easier for our partners. So that's why we have already in the last 2 years, we have reached more than 200 partnerships based on these sequencing-based deals. And this year, we expect that this number will be doubled. So I will introduce the RenMice for how to build using those the sequence to build a bispecific antibody and a bispecific ADCs. So here I just have it's a chart to show the trend of the antibody drugs. So we know the first antibody drug was approved in 1986, but I will say the first successful commercialization of the antibody drugs started in 1997 with the approval of rituximab. But for a long time, the antibody drugs approved by the regulatory agency is very limited until 2014. So we see a boom of the antibody drugs approved in the United States and Europe. So that's also the year of the PD-1 approval. But in the last several years, we see another trend. So besides the monoclonal antibody, we see more bispecific antibody or ADC drugs approved. These are also antibody-based drugs, but they are considered as a more complicated drugs. So that's why I will introduce how we can help with building the bispecific antibody or even bispecific ADCs. So to build a bispecific antibody, we need this tool. This is called the RenLight. So in this is a mouse that can generate the fixed light chain antibody. So what we did here is we fix the light chain with a rearranged VDJ sequence. But at the same time, we replaced the entire variable region of the heavy chain with a human sequence. So we are very unique in this mouse because we're the only one can generate a mouse with entire variable region of the human VDJ. Means this we put every single genes in the variable region in this mouse. So this cannot be done by any other companies, but because we are the expert in the gene editing technology. So we can do genome-wide megabase scale DNA fragment swapping. So that's why we generated such a fantastic mouse for all the antibody discovery. So with this mouse, so every time if you immunize them, you will generate the antibody sequence which you have this. If you look at the light chain sequence, they are identical, but the heavy chain sequence will determine the antigen specificity and also the affinity of the binders. So with this, the generation of building bispecific ADC antibody is much easier. So the middle one, you'll see the conventional way. If you generate the two different antibodies, if you put them together, you get a mixture of the antibody because the heavy chain, there's two heavy chain, two light chain. They will pair with each other by chance. But with our approach, if you have two different antigens in mind and you immunize two different cohort of mice and you generate two different pool of antibody. And after first round of selection, you will find the good binders for each target. Then you can co-express two different heavy chain and one single light chain in the same expression cells, and we'll assemble into like a bispecific antibody with very high purity. And we had the various tests, and here we just show the bottom. If you look at if we want to look at either by charge homogeneity, or if we conjugate with VC-MMAE payload, we can see typical distribution of the payloads, which is very similar to the monoclonal antibody conjugated with VC-MMAE. This means suggests this bispecific antibody really have very similar properties as a conventional monoclonal antibody. So this will make the production and also modification of this antibody, bispecific antibody, much convenient for all the users. So since it's a bispecific antibody, so people will ask a lot of questions about the technical things about whether this is readily convenient for the production. So here I show one example is the production yield. So this is expressing one of our projects like we did. Now I think it's already in the clinical trials. So after we generate a stable cell line, so we can get a very high titer. So this is like this is a bispecific antibody, but we can get an expression level at over 8 grams per liter. And if you look at either by SEC or by HIC, you see very very nice, like a single peak, means the product you get is very pure just after a single step purification step. So besides bispecific antibody, so this bispecific antibody, because it has a very similar structure as a monoclonal antibody, so easily you can conjugate with any, if you are working with ADCs, either by like sugar-based conjugation, cysteine-based conjugation, sortase-based conjugation, you can treat this molecule as easy as a monoclonal antibody. But here we also provide our unique linker payload system if you are interested in. So here, so we are building, this is our general structure for a bispecific ADC program. So for the antibody part, we have more than 200 TA targets freely available for your testing and assembly. And we also derived our unique linker payload system. So the payload is also belong to the topoisomerase inhibitor family, but it's highly potent compared with the DXd, and it also has very strong bystander killing effect. But the linker is also important because sometimes the linker is hydrophobic. It is not a very desired property for the drug development. Our linker is modified, so it's highly hydrophilic, and also it's cleavable. So it's very stable in the circulation and only cleaved once it gets into the tumor microenvironment. So this is the overall structure of our linker payload. So basically, we already filed a patent in last year. So this can be disclosed. So you can see that on the top, this is the structure of the linker payload. The red part is the payload, and we also have a linker, and which linker we have a side chain, so to ensure this the whole molecule is more hydrophilic. So on the bottom we show some the characterization of this system. So the left panel shows the in vitro killing activity. So we are comparing with the DXd. So we are using 40 different human cell lines. So we are looking at the in vitro IC50 compared with the DXd. Our payload is called BCP02. So compared with the DXd, you see the IC50 value is lower. So it means it's more potent than DXd. So this can be translated into the in vivo data. On the right, we're using a PDX model, and this is a tumor-bearing mice with a human PDX sample. So we conjugate the same antibody with either DXd from Daiichi or our linker payload BRD1102. The red line is from our payload. You see almost very good tumor suppression in this activity at this dose. But at the same dose, the trastuzumab conjugated with Daiichi's DXd show limited response. So this also consistent with the in vitro activity. And we also, in the middle, we show the whole molecule is very hydrophilic, because when the trastuzumab is conjugated with DXd, you see the shift on the reverse phase HPLC column, means the DXd from Daiichi is hydrophobic, which will cause the aggregation and the bad pharmacokinetics. But with our linker payload, even the elution time is the same as naked antibody. This is because the BRD1102, the new linker payload, is very hydrophilic, which is very good for the ADC development. So here I will just show you one case. So how we develop one of the assets using our bispecific antibody platform and our linker payload. So this one is called BCG033. So BCG is bicytogen. So this is the project number 33. So it's a bispecific, novel bispecific ADC targeting two different TA targets. One is PTK7, the other is CHOP2. So if you're working with the oncology, you should be very familiar with CHOP2. CHOP2 is clinical confirmed the target for ADC development. So far, there's two different companies already have their product approved, and both one is Immunomedics, the other is Daiichi, and both products is using the topoisomerase inhibitor as their payload. So this is a approved target. So, but even with that, it still have encountered some issues because the CHOP2 is widely expressed. So at high dose, it shows some toxicity, especially CHOP2 is expressed, also have expressed in the normal skin. And the other target I will introduce is PTK7. PTK7 is a relatively new target, so it's still at the earliest clinical stage research. But in the last couple of years, it has shown some very promising results. That's why we think there's a chance to combine PTK7 with CHOP2 to overcome the toxicity issue in the normal tissue by the approved CHOP2 ADCs. So this shows the early clinical data from for the PTK7. Although it's still at a very early stage, but it shows very good clinical response. So when we look at our database, so this is looking at the human on the left. You see the human human public database. We look at different type of tumor, and PTK7 and CHOP2 are both widely expressed, and they are co-expressed by many different type of tumor, including non-small cell lung cancer, breast cancer, ovarian cancer, and prostate cancers. And when we even further look in the literature, so this is shown on the right, we also see these two targets are co-expressed by many different type of tumors. So this is really encouraging for us because this seems like a good combination to overcome the CHOP2 single expression issues. And when we further looking at our internal PDX data bank, so we look at different PDX models. We see PTK7 and the CHOP2 are co-expressed by triple-negative cancers, ovarian cancer, lung cancer, colorectal cancer, esophageal cancer, and gastric cancer. So this is kind of like two good targets for many type of tumors. So here, what we think is we can build a bispecific antibody to overcome the CHOP2 toxicity issues. The rationale is here, like on here, you can see the normal cell express either PTK7 or CHOP2 at a very low level. So if you're doing a monoclonal antibody ADC, you're not likely to differentiate tumor cells from the healthy cells. But the tumor cells, since they co-express two different targets, so we design a bispecific antibody recognize both targets. So the antibody, bispecific antibody, will bind to the tumor cell much better than a normal cell, which only express one target. So this is one way to increase the therapeutic window. So what we did here is we generate the CHOP2, select CHOP2 and the PTK7 arm for bispecific antibodies, and we further conjugate with different linker payloads. With either initially we are doing that with VC and MMAE. Then we try with our new linker payloads to show the performance. So selection of the bispecific ADC for us is like this is very routine for our internal campaigns. So we first immunize the different mice and with the two different antigens, and we generate a collection of binders. Then we are going to a series of in vitro selection criteria, like whether they have good affinity, they bind to different species, and we also check the developability, like physical chemical properties, to ensure only the molecule with no defect can go into the next step. Then once we have usually we come with 3 to 5 different molecules for each binder. So we are doing the internal assembly into a bispecific molecules and further conjugate with different linker payload and test doing the in vitro and the in vivo testing. So this shows the data. So both the PTK7, this bispecific antibody combined to the human CHOP2 and the monkey CHOP2 and the human PTK7 and the monkey PTK7. And we are always using all the benchmark from the 2 approved drugs for CHOP2 and the 1 clinical drug for PTK7 as a benchmark, show the binding specificity and also using that for all the functional comparison. So this also shows the bispecific antibody combined to a variety of human tumor cell lines. So this is we also show that they have the binding affinity binding is as good as all the benchmark. So this is the in vitro assay shows like for different tumor cell lines, they express different level of PTK7 and the CHOP2, but the bispecific antibody, which is showing the red color, can bind to the tumor cell very well. And for ADC development, we really care about the internalization, because this is a way to release the drugs in the tumor cells. So we also compare with the the parental antibody of PTK7 and TROP2, the bispecific antibody, which is shown in the red color here, also show very nice internalization within 6 hours. So we further move down to the in vivo assay using the different PDX models and the CDX models. So here we compare with the bispecific ADC with the parental. On the top is the compare with the parental monoclonal antibody ADCs. On the bottom we compare with the benchmark ADCs. We see very nice tumor inhibition in different tumor models. But at this stage we are conjugated with VC-MMAE for proof of concept. And further, since we already have derived our own linker payload, so we switch everything to the new linker payload system. So this is the final structure of the BCG033 bispecific antibody with our unique linker payload. So here we also show with the new linker payload, we are doing that comparison in different PDX model with the parental monoclonal antibody of PTK7 and TROP2. We see very nice tumor inhibition regardless of the expression level of two different antigens. And we also compare with the benchmark, either the TROP2 Dato-DXd or sacituzumab conjugated ADC or the cofetuzumab conjugated PTK7 ADC. We see very nice tumor inhibition, which is in most cases our bispecific ADC show much better performance compared with the benchmark. So one interesting thing I want to share is here, so show the comparison of the treatment of BCG033 with all the 3 different benchmarks. So you see after one single dose, our BCG033 bispecific ADC can suppress the tumor growth, but all the 3 controls, they are not very effective. So it means they are resistant to this type of tumor is resistant to these benchmark drugs. But so we separate those mice and drug-resistant mice into 2 different groups. And one group we give 1 additional shot of BCG033, and then at this time we see the tumor volume goes down. Means even those tumors are resistant to the benchmark ADCs. They are our linker payload conjugated BCG033 is very effective in these settings. So overall, I think this is one example by surgeon data for derived bispecific ADCs. We rationally select 2 different targets for targeting PTK7 and TROP2, and this has been a very useful combination and very promising for a variety of tumor type. And we showed in vitro and in vivo, especially in vivo, we see much better performance compared with the parental monoclonal antibody ADCs and also other benchmark ADCs. And I think this will be a very good asset for clinical trials. And overall, I think because we have a very powerful platform to building on really building bispecific antibody. So in the last several years, we have generated a series of bispecific ADC assets, and some of the assets are with our partner. They already started the clinical trial, phase 1 trials. So we have 3 assets on top of this slide. They already entered the phase 1 trials in the United States, and we also have several other assets. We have external partners, but overall we still have a lot of new assets we are generating currently. So besides what we show on the last slide, we can really building novel bispecific antibody using our antibody libraries. So we have antibody binders for over 200 TA targets. So this can be easily using new molecules using a plug and play approach. So we can our partner can nominate some combination of ideas, and we can quickly building a bispecific antibody and for further downstream testing. So besides the common light chain approach, we can also building the multi-specific ADC with the nanobody derived approach. So this is a conventional way we're building bispecific ADCs. But now we have another platform called the RenNano. So RenNano is a transgenic mouse that can produce fully human nanobody or heavy chain only antibody. So this is much flexible because this time we don't need to worry about the light chain pairing. And then the nanobody is also smaller, because so they can easily be put together to building a larger multi-specific molecules. So currently we're also testing using the nanobody to building multi-specific ADCs, like putting on the backbone of existing molecule. We can put a different nanobody targeting a different TA targets or targeting VEGF and building a tri-specific molecules. Or we can put it like on the bottom, we can also building another ways of nanobody, like putting multiple nanobody in tandem repeats. We call nanobody string. So this is another way to building multi-specific antibody, or then we can conjugate with different drugs. So I think overall, I think BioCell have developed a very powerful platform for the antibody discovery, and this can including monoclonal antibody, bispecific antibody, and nanobody for without light chain. So we have been already established a library of over 1 million fully human binder sequence. So these binders are readily available for our partner to testing and also for licensing. And our technology has been really both the technology and our antibody sequences have been widely accepted by a lot of companies, and that's why I think we are the company have a large number of collaborations and with the global biopharmaceutical field in the last couple of years. And if you're interested in this technology or the binder sequence, you can let us know, and we can provide you more detailed information for your consideration. And I think that's my introduction for RenBiologics. And now next I would like to let my colleagues to introduce the other part of BioCell's business, the biomice. If there's no question, we can switch, and then we can probably have the question and answers in the end of the session.
Hi everyone, nice to meet you. This is Sha. I will give you a small introduction about myself. So I just joined BioCell like 2 years ago. So I'm working in the pharmacology department and working as a senior product manager and also principal scientist. Today I just give you a presentation about the crossing the BBB with the preclinical evaluation methods that we have in our site to kind of give you an idea what we can do in our site. And yeah. So today's content is about, we will walk through the structure and function of the BBB first, and then I will introduce the key preclinical evaluation methods and also the advanced neuro-related method models that we have. And last, I will give you a summary and future directions that we use in our site. Okay. Okay, so I think everyone should know something about the BBB, and so let's look at the BBB. So which is a semi-permeable barrier that regulates molecular exchange between the brain and blood. So here is the details about the BBB structure. So which is constituted by different cell types. For example, the microglies in the brain. For example, the endothelia. So we can see in the middle is the blood vessel, and outside the first layer is the endothelial cells. In the endothelial cells in the brain, they will connect it to each other to build the tight junction. And the next layer is the parasites. Okay, so so this is the structure of the BBB, and so it prevents the it helps to protect the brain at the same time, but it prevents most of the drugs to cross the BBB. And due to the urgent needs that because there are so many patients, they have like more. Because the aging population, many people can live longer, so we have much more diseases that are related to BBB, and also the key like technical issues that already there. So we need to we had the urgent to do something to find a more better way to do the drug discovery for crossing the BBB. Okay, so yeah, most drugs fail to cross the BBB, and then we have the urgent need to do the brain-targeted therapeutic to cross the BBB. So in the brain, there are multiple pathways to let the drugs across the BBB. The first one is we can use in like lipid soluble or water soluble way to let some of the drugs can cross the BBB, and also we have the receptor-mediated way transcytosis. So where the drugs can bind to the receptor on the endothelial cells and then build up a vesicle and then transit to the brain. And other ways like cell transcytosis and also the lympholytic pathway, which is related to maybe some lipid nanoparticle, and also we have other pathways. So these are all the pathways that can help the drugs to cross the BBB. And to help the drug successful cross the BBB, there are already some strategies that people are doing. For example, people have tried using different ways for dosing the drugs through the nasal. And also, I just mentioned we can also using different nanoparticle delivery system. And And today I just want to highlight the receptor-mediated transcytosis. So here we can see is the receptor-mediated transcytosis, the typical receptors here. The first one is the transferrin receptors. So, which is, it is most widely used and validated receptor for the drugs to cross the BBB. And also we have the CD98 heavy chain, which is also important, and I think recently this receptor-related transition is also very promising. And also we have the LDLR-related receptor and insulin receptor, but all these are related to for example, the insulin receptor, it only can transfer the insulin and other related ligands across the BBB. And in biocytogen, we do have a lot of humanized mice, if you know anything about our company. So we have developed many like animal models that the target has been humanized. So here is a list to show you the animal models that we have. For example, the TFR1, we have all those humanized mice ready to use. And for each of the receptors, we also developed many humanized mice. So just check our website to find if there are any strain that you are interested in. You can find any data, in there. And so the key preclinical evaluation method for BB penetration here in our site, I will just take TFR1 as an example. And also we have a lot of like case study or experience in this strain too. First, this is just a general information about the TFR1 mouse. So we can see that we have compared our TFR1 mice versus other mice from other vendors. We can see that our TFR1 did have all extracellular domain is humanized, and also we have a lot of experience doing this, and so we have tried different models in our site too. And this strain is also very popular in the last a few years, last year. So it has a monthly sales like exceeding 1,000, so which is really good. And this is expression data to show the human TFR1 mice to let you know we have done like a lot of validation data for each of the humanized mice. So basically, we will have like RNA expression, protein expression, and the expression in different organs. Just to give you an idea, we have a lot of validated data package for each of the product. And also for this specific strain, there are many researchers have using the strain to publish a lot of like beautiful work. And yeah, this is also another ad, I guess. So here is a preclinical. Here is the like a workflow to show you that what we can do in our site. So in general, the drug will be administered through different routes, and we will analyze the animal and then collect the blood sample for PK study if you want. And then we also can collect the CSF samples. As you may know, the CSF samples is really hard to collect, and for the mouse strain, because it's too small, the limited amount of CSF is very precious. So we have a lot of experience doing that. So you can see the imaging in the right corner, and that's the CSF collected from the mouse. And actually, there is a movie. Never mind, I cannot move my. So there is a movie over there, so which shows that the CSF like flowing outside the right. So because for the mouse, we can only collect a limited amount of the CSF, but for rat, we can do some like operation to introduce a tube into the mouse brain and then collect the CSF multiple times. So which give you a way to collect the samples from different time points and do the PK study. And that's for the CSF. And then we can do hyperfusion for the animal and study, and then collected the brain for PK study or collected the brain for the histopathology analysis. So this is just an image to show you that we have the experience to collect different regions of the brain according to your needs. And then we can do related detections after that. And this is a case study to show you that, we have using the human TFR1 mice and to do the dosage for one drug, and then collected the brain and to do some, to homogenize the samples, and then to centrifuge it with our protocol, and then collected the samples from like top and middle or bottom samples. Then we can separate the tissues to parenchyma or transparent middle part. I think it's just junction. And bottom is capillaries, CD31, positive. And also we can do PK study for this one. So this is just a data to show you that we can collect the region from the brain by different, for example, the whole brain we can analyze, and also the parenchyma and serum. We can do the PK for each of the region, and by using our platform, so where we have the ELISA and ACL, of course. This is another case study where we have using our TFR1 mice and to test the bispecific antibodies. So here we can see after dosing the antibodies, we can see the ELISA binding to the TFR1. And also we collected different region of the brain and to do the like PK study too. And in the left, we also can see with the bispecific antibodies, they have higher penetration into the brain compared to the control group. And this is also the PK study where we collected different samples from the brain, where we can see it is very consistently with the TFR bispecific antibody, but it decreased in the serum. So which means the bispecific antibodies can penetrate to the brain very well compared to the control antibody. And apart from the PK study, we also can using our platform to do the biodistribution analysis. So this is just an example to show you that we can collect the sample and using the imaging system to capture the intensity of the test drug after dosing it. And another humanized mice I also want to mention is the CD98 heavy chain. So for this one is slightly smaller amounts compared to the TFR1, but it's still very popular. So just give you an idea, this is also a choice. So similarly, we also did like validation data for this strain too. We have do the mRNA expression, protein expression, and also the immunofluorescence to see the locate in the brain too. And yeah, the third part, I will go in through all the advanced neuro-related models that we have in our site. And yeah, so generally, we can classify our animal model to 3 parts. So the first one is cancer-related. So where we have the brain cancer animal model. And the second part is biomass-related. So where we have like a target related that to neuron science related disease models. So the third one is the induced in neurologic disease models. And this is the first one. So where we can using our site to establish in the orthotopic brain cancer model. So this is to show you how we did that. We can using the injection of the tumors into the brain, and then using the IVIS imaging system to real-time monitoring the tumor growth over time. And also we can using our micro CT to do this real-time monitoring too. And we can see, the tumor is growing bigger and bigger over time. The next part is the biomass animal models. So actually for this part, we have done or established a lot of different humanized mice, which including like AD, ALS, PD, SD, too much. So today I just choose a few of them to show you the data we have. So the first one is the Alzheimer's disease. So which is characterized by the accumulation of the A-beta plaque. So and also it will processed with the tau protein deposition, which were related to the cognition irreversibly. So which is really and there are like few risk factors that are related to this AD model too. So here we have highlighted that the first risk factor is PSEN, which will cause the most severe forms of AD. And also we have APP mutation, which causes an increase in the A-beta-2 and A-beta-40 ratio. And so based on this, we have actually made a mouse strain, which is APP and PSEN related mice. So in these mice, we have using the mutation. So I don't know how they did make the targeting strategy, but what I do know is they're using the mutation way and to make the humanized product. So this is the targeting strategy. So where we can see that in this strain, so this is actually a product after crossed by the APP mice and PSEN mice. So this is a targeting data. And also we can see the A-beta expression in the by histomatic. And also we can compare to the astrocytes, which we label with GFAP, and to see the protein expression in this model. And another neuro-related model is PD, so Parkinson's disease. So Parkinson's disease is related to the loss of dopamine producing in neuron cells, and also the build-up of new Lewy bodies. So here we can, it also will affect the motor or non-motor symptoms. And this is, there are so many risk factors that related to PD animals. So we have, actually, I think we have more animal models. I just choose one of them. So for this specific gene, which is related to the misfold and aggregate into the toxic Lewis bodies. So we have also made this humanized mice where this is the protein expression data. But for this basic one, if you may notice that there is a leakage of the SNCA in different organs compared to brain only. So for this specific strain, we actually did a plus version. So in this plus version, we can see that it only expressed in the brain, so which is pretty good. And since the Parkinson's disease is related to the behavior a lot, we also have our behavior test platform for you to choose to evaluate the behavior of the animals. So we can see here we have establishing the movement fatigue behavior test. The green label one are the available behavior test. But with the green label one, the test is still like under development. So we will keep going to make the evaluation system much more, much more better. So if you are interested in any of the details data, you can also let us know. So we also establishing the emotion and cognition learning and memory part for the evaluation part, and also the perception part. Yeah, and also we have the scratching test part available, which is also used for the autoimmune disease models like pruritus. Yeah. And yeah, this is an example to show you the behavior test after evaluating the PD mice, which is HSCNA mice. So we can see that by using the behavior test, we did see this abnormal behavior in the humanized mice. So we compare the by open field test and also the rotary test in these two different, and also grip strength as well. And we compare between the male and female too. And this is just the modeling data, so we didn't add alien drug here. And another disease is ASD, autism spectral disorder. So this disease is really linked to connectivity and synapse disorder, so which also linked to the genetic and development for early, and also varies between individuals a lot. And there are also a lot of risk factors in this model too. So for introduction, we have here listed two of them. One is FMR1 knockout mouse, and the second one is MECP2 knockout mice. So actually, we can see that we still have some data that to be collected for this model, but we just want to show you that we do have many studies ongoing. So for this specific strain, where we have already done the protein expression, and we will do more behavior tests to show if this knockout mouse did well affect the or develop the ASD afterwards. Ooh. Okay. And the next one is it's another strain. So where for this one, we also did the protein expression analysis, and we have using the open field test to measure the behavior in this strain, where we can see that it has a normal movement compared to the regular wild type one. And also, these are the other way to measure the pain. So I'm not sure if you are interested in the pain model too. So we have established the multiple pain model too. For example, the mechanical evoked pain and heat evoked pain and cold evoked pain, and some other pain are just under developing. So we were using the von Frey testing to test the pain. And these are just a case study to show you. The left part is a CFA-induced inflammation pain, where we did CFA injection on day 0 and did the von Frey testing on different time points. Because for the pain, we need for all those behavior, we need to acclimate the animals for a decent amount of time to make them quiet or not affected by the environment. So we have establishing all those methods to do the pain evaluation. So the right part is the B6F10 cells. I think it's B16F10 cells, induce a cancer pain. So after injection with them, we also did the von Frey testing on different days. So the last part of nervous system related disease is induced disease models. So since we have a lot of autoimmune disease models, and this one is just part of them, for nervous system, we have established in the EAE, which is a multiple sclerosis model. And in this model, so we have induced with MOG. And this data also shows a drug efficacy where we dosage the PTX at day 0 and day 2. And in this model, we have, so for EAE, we did have a clinical score to evaluate the disease progression. And also we will have histopathology analysis to see how the drug efficacy as well. And from this data, we can see that the modeling is pretty successful. And then the drug did have a inhibition of the disease progression. And this is another case that to show you the MCAO model that we have. So because if you know that this also is a very delicate operation, so we need a very experienced operators to do this study. We want to reduce the variance between person or between the animals to give you a more stable data when you're using this animal. So we have tried to establish in this model too, where we can test the MCO region and also the behavior of the animals too. This is the behavior where we have using the rotary test to see the behavior and where we can see the compare. So this is only modeling data where we did see after modeling the behavior decreased a lot compared to the sham operation group. And also, to help us to monitoring the behavior very well, we have established a lot of cages with a camera on there. So we don't need to like monitor the animal at a specific time. We just turn on the camera and the animal's already, behavior has already been monitored. So we will have the results. And yeah, I think, yeah, this is also, I don't know if we can play that. Oh, it can. So we kind of have a camera on there and monitoring the behavior of the animals. Yeah. Who count? Yeah. Yeah, so after collecting the video, yeah. No, still labor. Yeah. Yeah. And yeah, that's all the typical neuron-related models that we have so far. And many of them we didn't show that because of the limited time. So today I just want to give you a brief idea. So the key takeaway I want to emphasize a lot is first is the TFR1 mice. So which is really the most popular strain to help transfer to the brain. And also we have CD98 heavy chain mice, which is really a rising star in the BBB transporting. And also we have a well-established pipeline to analyze the drug penetration into the brain. So depending on different samples or different detection that you want to do, we have established methods for each of them. And the next one is, we have 3 different types of neuron-related models, the brain cancer and the biomice, and also the induced neuron disease animal models. And apart from that, we also have multiple detection methods that can be used to evaluate each of the model. For example, we have real-time monitoring system with IVC imaging system or microCT. We also have behavior testing to evaluate the biomice behavior. And also we have, a lot of induced neuron-related model, and they're included a really sophisticated surgery there. And I think that's it. And also I want to give you an introduction about our biomice. So our biomice included the gene editing animal model generation, which means we have the gene editing service. So if the animal that on the shelf is off the shelf is not your needed, you can just let us know. We can do the customized gene editing for you, and then to establishing the mouse models. And also we have the large scale animal model supply, which is really important because it will keep us like maintain all those studies going and maintain all those animals could be available at very problem time. And also we have our pharmacology service based on all those mouse models. So all the 3 parts just keep us biomice ongoing. So our biomice, have already have over, I think this number, it's correct. So it already has over 4,900 customized projects delivered, and also we have over 3,300 off-the-shelf humanized mouse. And also we have the pharmacology service based on different animal models. And partnership, we have a service for all the top 10 MNC using our mice. And for the future, I already showed you this list. So actually we have more of the each of the disease models. So if you would like to check if of each of them, there are a lot of slides you can get, hopefully. And also, I think as a plan, we are still going to make a lot of humanized target animals at the same time, and we'll generating different animal models based on all those mice and transfer to the humanized models too. Also, we will establish a better evaluation system for each of the behavior. And hopefully we will get the, for example, the BBB evaluation preclinical model, much more contents next time. Yeah. And yeah, this is a complete. I think Dr. Yang has already introduced a lot. So that's all. Thank you.